Hi guys, so today I'm going to be teaching you about a quick little overview of cell division mechanisms. Now, the continuity of life depends on reproduction. By this process, parents produce a new generation of cells or multicelled individuals like themselves. Cell division is the bridge between generations. So during cell division, a cell faces many challenges. Each daughter must get information encoded in the parental DNA and enough cytoplasm to start its own operation. DNA tells what proteins to form. Proteins are structural materials and others are enzymes. If a cell does not inherit all of the required information, it won't be able to grow or function properly. Inheritance of cytoplasm comes with organelles, enzymes, and metabolic machinery for the point until it can use information in its own DNA for growing on its own. Mitosis, meiosis, and prokaryotes. Eukaryotic cells can't just split into two. Their DNA is housed in a single nucleus. So how do you expect something to make an identical copy of itself if all of the genetic information is stored within one compartment? That's the magic of cell division. So they do split the cytoplasm into daughters, but not until after the DNA has been copied and packaged into more than a single nucleus by way of mitosis or meiosis. Mitosis. It's a nuclear division mechanism. It occurs in somatic cells. Only multicellular eukaryotes undergo mitosis. Mitosis is the basis of asexual reproduction in plants, animals, fungi, single-celled protists. Occurs to increase in body during growth. That means increase in size during growth. Also, mitosis occurs to replace dead or worn out cells and repair of damaged tissues. So, meiosis. Meiosis is also another nuclear division mechanism. It precedes the formation of gametes or spores and is the basis of sexual reproduction. Gametes are sperm and egg cells. They develop from germ cells or immature reproductive cells. Spores form in the life cycle of many protists as well as plants and fungi. So the key structure of a chromosome. So eukaryotic chromosomes are one double-stranded DNA molecule with a lot of proteins attached. Each species has a different characteristic number of chromosomes. Before a cell enters division, it copies each chromosome. A copy attached to the original chromosome stays attached as sister chromosomes. So we have one unduplicated chromosome, which is called a chromatid, and one whole chromosome that's duplicated, and that's called sister chromatids. And we'll further in the video, we'll go into what these structures are. So during an early stage of meiosis or mitosis, each duplicated chromosome coils back on itself. A duplicated chromosome in its most condensed form is shown right there. The coiling arises from interactions between the DNA molecule and proteins associated with it. So how does loose DNA become a chromosome? It's all in the turns. At regular intervals, the double-stranded DNA wraps 2 times around tiny spools of proteins called histones. This structure is called a nucleosome. So this structure that looks like a bean, that's called the histone, and that's the DNA wrapping around it twice. And this whole structure makes a nucleosome, So several nucleosomes almost look like beads. They're the smallest unit of structural organization in eukaryotic cells. While each duplicated chromosome is condensing, it becomes constricted. This constriction is called a centromere. The location of the centromere is different for each type of chromosome. The centromere is a defining characteristic of a chromosome. During nuclear division, we find a kinetochore at the centrum of each chromatid. The kinetochore is the docking site for microtubules that will help move chromatids during nuclear division. So that whole center region in a chromosome is called the centromere, and within a chromatid, that outer region basically of the center of a chromatid is the kinetochore. So what's the point of this structural organization? The tight packing keeps the chromosomes from getting tangled while they move during nuclear division. Between nuclear division, the nucleosome packing loosens. So basically, it starts off as loose DNA, and that's a histone. So the loose DNA coils around a histone, making a nucleosome. Then many nucleosomes get attached to more DNA, which makes chromatin. And that's what loose chromatin would look like within a cell. Then the chromatin gets coiled up and it becomes a chromatid. And that outer structure on the chromatid is called the kinetochore, and that's an unduplicated chromatid. Once the chromatid gets duplicated, it becomes a chromosome. And that center adjoining region of two chromatids is called a centromere. It's a lot of C's, but it makes sense after a while. So summary. Eukaryotic cells divide their nucleus first, then their cytoplasm. Mitosis is the basis of bodily growth, cell replacements, tissue repair, and often asexual reproduction. In meiosis, the formation of gametes, and in many species, spores, is what precedes nuclear division. Gametes are sperm and egg cells. Meiosis makes non-identical cells. Mitosis makes identical cells. Meiosis comes after interphase, and it's the reductive division of cells. In meiosis, PMAT occurs twice, and if you watched my other video on mitosis, you'd know what PMAT means. So basically, meiosis is after nuclear division. So first nuclear division, then formation of gametes and spores in meiosis. Thank you guys for watching, and feel free to watch my other videos.